So let's talk about osmoregulation in plants. This is the syllabus objective, is to explain how structural and homeostatic mechanisms maintain water balance in plants. We need to include the roles of the stomata, the vacuoles, cuticle, and abscisic acid, ABA. So here's a couple of different plants just to show that there's a whole range of different adaptations that support plants to be able to conserve water. So this is the curly sedge, and the curly leaves tend to trap the morning dew. So here's the grass tree. It's got very small surface area of the leaves, and the leaves run into this centre. So when it does rain, the rain runs down the leaves and down towards the base. So this is a rainforest plant. It doesn't have any issues with losing water. This is the issue here, is that it needs to have lots of sunlight, so it has really large leaves to be able to exploit that. And what you can see on here, this is salt. So this is a mangrove plant, and because it lives in salt water, it excretes the salt out of the plant, and one way it does that is through the leaves. And here's a hydrophyte, a water plant, and an adaptation that it has. Again, it doesn't really have any issues with losing water, but it has its stomata on the top surface of the leaves. And here's the cactus, which doesn't have leaves at all, and it actually photosynthesizes through its trunk. So different plants living in different environments have different adaptations. Okay, so let's look at osmoregulation in plants. Water enters the plants through the roots, and it is lost through the stomata. Now, we've talked in previous videos about how the mechanism of water evaporating out of the leaves and transpiration is not only is it the way the plants lose water, but it's also the mechanism by which water is pulled up through the plant. So it's kind of like a double-edged sword. But importantly, this is how plants can control osmoregulation through the stomata. One of the other syllabus objectives was to look at how different factors can affect the rate of transpiration. Light, when there's high light, there's more transpiration, because when there's more light, there's more photosynthesis, and that requires the gas exchange. High temperatures, there's greater gas exchange. High temperatures, there's greater evaporation or transpiration, because particles are moving faster when there's more higher temperatures, so there's more energy, more evaporation. High humidity actually reduces the amount of evaporation or the amount of transpiration, because the air is already saturated with water. High wind, high evaporation, so greater transpiration. When there's high soil moisture, then transpiration can occur, no problems at all. But when there's low soil moisture, we're going to see how the stomata close, reducing the amount of transpiration. So, firstly, we need to have a look at the structure of the leaf specifically related to osmoregulation. So this is a cross-section of a leaf here. Of course, the leaf is the functional unit in which photosynthesis occurs. And you can see these little green circles here. Well, these are the chloroplasts. And so the area in which photosynthesis primarily occurs is in this palisade layer here, where you've got these vertical cells that are stacked next to each other, increasing the surface area for being able to capture the light. But above that, we have an upper epidermis, which protects the leaf. And that is, can you see how flat those cells are? And that allows light to travel through maximally. But above that, we've got the cuticle. The cuticle is waxy. It is transparent, allowing light to travel through, but it's also waxy and it's waterproof, so it prevents water loss through the top of the leaf. Now, if we look down here in this next part, we've got what we call the spongy mesophyll. And what I think is really cool about this is that it's got all of these large air gaps and a large surface area. So these air gaps allow an exchange of carbon dioxide and oxygen between the different cells without having to exit the leaf. So this can occur while the stoma is closed. And then this next part down at the bottom of the leaf, normally we see the stomata or the stoma at the bottom of the leaf to avoid excessive water loss. So we're going to look at the stoma in a minute, but as you probably remember, the guard cells are the cells that control the opening and closing of this hole called the stoma, and it's through this hole that we have gas exchange. It's also the hole in which water is lost through transpiration. So we're going to talk about the stomata in just a moment, but the other thing we need to talk about was the vacuole. The vacuole is helpful for storing water inside the cell, of plant cells. In hypotonic situations, so when there's low solute and high water outside of the cells, water enters the cell and makes the vacuole swell. This is the situation where plant cells really like, because the water pushes against the cell wall in a condition we call turgor, and this turgor pressure maintains the rigidity of plants. So you imagine if you forget to water your indoor plants for a while, they become very floppy, whereas when you water them, the water goes into the cells, creates this turgor pressure, and it maintains the rigidity of the plant. In hypertonic situations, water exits the cells, so the vacuole shrinks, and it actually falls away or retracts away from the cell wall, and that's a condition we call plasmolysis. Plants much prefer a hypotonic situation to produce that turgor pressure. So now let's have a look at the stomata. Okay, so stoma is the individual or the singular, stomata is the plural, and a leaf has hundreds, if not thousands, of stomata, and they're controlled, each of them is controlled by two guard cells. So the guard cells open the stoma when the guard cells are swollen. Now, the way this occurs is that actively ions are pumped into the vacuole and water then follows. So the guard cells swell, and because the inner side of the inner cell wall of the guard cell is thicker than the outside, when the guard cell swells, it ends up with this jelly bean shape here, and on either side, it opens up a hole in the middle. So when the guard cells are swollen, the stoma is open, and that allows gas exchange, but it also allows water loss and transpiration. When the guard cells are shrunken, the stoma closes and no water loss occurs. So the guard cells are able to control water loss through the leaf. The issue, though, of course, is that when the guard cells close, no photosynthesis is going to occur because there's no gas exchange and provision of carbon dioxide. Now, so the other syllabus objective was to talk about abscisic acid, or ABA. It is like the stress. It is like a stress hormone in plants. And I've just got here about xerophytes. Now, xerophytes are plants that live in dry conditions. So plants that live in dry conditions are under water stress. So one of the ways that plants can respond to that is by releasing this stress hormone called abscisic acid, and it then makes the abscisic acid then influences the stomata to close. Okay, again, it's about through the movement of ions. Movement of ions out of the vacuole means that water follows and it becomes flaccid and the stomata closes. So